This is a grey fluid. How exciting. Secretly, inside this little beaker, a very exciting process is going on. Everywhere in that beaker, gold ions are forming big groups and interacting with each other, kind of like one big group hug. Or rather, a bunch of cars crashing into each other. Nanoparticles can be used for a lot of applications, from imaging in the medical field to preventing you from getting smelly feet. Gold nanoparticles in particular show some very interesting optical properties. A lot of plants show the ability to take up metals. Every living organism needs some metals to survive, but sometimes we can hijack the uptake mechanisms of these plants to gather other kinds of metals. Gold is one of these, and when you expose plants to a solution containing gold, they'll nicely gather it for you and store it somewhere in a nice, compact fashion. Nanoparticles. We decided to try a very low concentration of gold tetrachloride, around 10 milligrams, and use mint plants to see if nanoparticles would form. This concentration has previously been shown to influence plant growth, but it's never tested whether or not nanoparticles would form. So let's start by cloning some plants. Why clone plants, you ask? Well, different plants may have different genes or different expression levels of those genes. So in order to rule out any genetic factors in how the nanoparticles would form, we decided to just use the same genome for them all. So cut off some mint, dip them in some root-forming hormones, and give them some water and soil, and soon we'll be able to harvest their organs, nanoparticles. Rinse and repeat, and soon we'll have enough plants to perform our experiment. Buy some lamps, and your mint will grow faster than you can say gold tetrachloride. We made a little setup to hold the lamps in the camera and left the plants to grow. After a week, they had formed enough roots to start our experiment. In order to ensure that the plants can grow healthy, we used a two-part hydroponic nutrient solution, creatively named Hydro A and Hydro B. The nitrogen, phosphorus and potassium contents are as follows. We used the manufacturer's recommended amount of Hydro A and B per liter. We also added 10 mg of gold per liter. In order to keep the experiment cheap, we used plastic bottles as reservoirs. We then add the plug with the mint to the plastic bottles. And now, we wait. Each week of the experiment, we made 2 new containers and added mint and gold to them, so that the plants would be exposed to gold at different sizes, to see if there was a difference. Every week we also took a sample from the growth solutions to analyze by UV-vis spectrometry. Fast forward a bit, and our plants are now 5 weeks old and are all grown up. Hooray! To celebrate, we made sure to dismember them. We split our corpses into 3 main parts: roots, stems and leaves. We then crushed them thoroughly with liquid nitrogen again and again and again. After crushing our souls, we were now ready to put our samples into a transmission electron microscope. The roots from our first plant showed clear nanoparticles, just like the roots from week 2. The leaf and stem samples, however, showed only organic materials. We measured the samples we took in a UV-vis spectrometer, and from the absorption spectra it became clear that after 1 week, pretty much all the gold had left the solution. Of course, we also ran into some problems. I'd like to introduce you to our favorite problem. We named him Arthur Bidopsis. After Arthur, other pests started showing up. Little mites covered the stems of our plants, and some strange black substance formed at the bottom of our bottles. Lucky for us, it seems like this is normal, as when our supervisor tried the same experiments, he also got black muck where gold was present. Because of this, we believe it's just some precipitated gold. During the experiments, we not only encountered pesky bugs, but some other organisms had also made a home of our plants. Mold. Lucky for us, not every bottle was infected. Conclusion? Mint will form nanoparticles in its roots when exposed to 10 mg per liter of soluble gold. However, these particles are not very well dispersed, as they range from 10 to 30 nanometers in diameter.